Welcome back, everyone, for our second day of our symposium facing up to disparities in healthcare. My name is Jason Torres, and I'm a junior research fellow with the Oxford Center for Personalized Medicine, or CPM. The CPM is, in turn, an outreach vehicle based at the Wellcome Center for Human Genetics and St. Anne's College. We are funded by both the Wellcome Trust and the Stanley Ho Medical Foundation. And the aim of the CPM is to promote a deeper understanding of both advances and challenges within the field of personalized medicine. We organize events throughout the academic year, including this event, which has been co-organized along with Dr. Jenny Douglas, who is a senior lecturer at the Open University and a Plumer Visiting Research Fellow at St. Anne's College. And we're very grateful for Dr. Douglas's expertise in helping us put this event together. The focus of this symposium has been to explore biases within healthcare systems that result in unequal outcomes in disease diagnosis, treatment, and prevention. Yesterday, we heard from expert speakers who discussed the unequal impact of COVID-19 on specific communities. We also heard from Professor Marian Knight, who discussed disparities in maternal and infant health, and from Jenny Douglas on the troubled history of the reproductive health of Black women in the United Kingdom. Today, we will start by addressing disparities in the field of human genetics. Before we begin, just a few housekeeping points though. Full biographies for all of our speakers and the full program can be found in the booklet that accompanies this event, which is linked from the email reminder that you will have received. We encourage you to tweet about this event unless our speakers specifically opt out. Please use the hashtag CPMHD21. Also, and importantly, please submit questions via the Q&A function, and we will aim to address them during the discussion sessions. Also, just another note, and unfortunately, Dr. Jeremy Farrar couldn't join yesterday, but we have been in touch, and he will give a presentation at a future date, so we will keep people up to date about that. All right, so with that said, I will now start session 3, which is titled The Importance of Equal Representation in Genetic Research. And as an opening remark, I think it's important to point out that since the completion of the Human Genome Project nearly 2 decades ago, geneticists have identified tens of thousands of genetic associations involving hundreds of complex traits and diseases, such as cardiovascular disease and type 2 diabetes. The results gleaned from these investigations have provided biological insights into disease processes that inform the efficacy and development of therapeutics. As such, human genetics has become an increasingly valuable component of drug discovery programs for many pharmaceutical companies. Moreover, genetic risk scores are showing utility in identifying patients at higher risk for developing particular diseases and that may benefit from earlier screening or intervention, or that may respond better to certain medications. However, the vast majority of genetic studies have been restricted to populations of European ancestry, which has created a blind spot for uncovering genetic risk factors that may be more pertinent in non-European populations. This imbalance poses a concerning potential to amplify rather than equalize current disparities in our healthcare systems. Moreover, the commodification of DNA information, which supports a lucrative industry of direct-to-consumer DNA and ancestry testing kits, raises additional concerns among marginalized communities that have been historically excluded from the benefits afforded by the study of their own genetic information. These are serious concerns, and I am delighted today to welcome a panel of exceptional speakers who have thought very long and hard about these issues and are actively working to address them. Our first speaker is Professor Emir Kenny. Emir is a professor of medicine and genetics and the founding director of the Institute for Genomic Health at the Icahn School of Medicine at Mount Sinai in New York. She leads a multidisciplinary team of geneticists, computer scientists, clinicians, and other medical professionals, working on problems at the interface of genetic ancestry, massive scale genomics, and medicine. She has published over 100 papers in leading journals such as Science, Nature, and the New England Journal of Medicine, with over 15,000 citations, and her work has been featured in many media outlets, including the New York Times. With that, I am very pleased to welcome Professor Kenny.
Thank you very much for that wonderful introduction, and I'm so pleased to be here today. I'm coming to you from New York City, where it's approximately 8:40 in the morning. Bear with me as I fumble to get my slides there. Okay, can you see them?
Yes, thank you.
Wonderful. Okay, so it's wonderful to be here, and this is, I think, a group of very like-minded individuals who agree that our lack of diversity and representation, not only in genomic research enterprise, but also in how we're thinking about using genomic medicine in arenas of medicine or public health, is really concerning. So I'm going to talk a little bit this morning about some of the work that we have been doing on this topic in the Institute for Genomic Health in Mount Sinai. And before I start, I just want to show my disclosures. But I really wanted to take a step back and think about genomic technology writ large in society. This is data that was announced in the AGBT meeting at the end of 2019. Lumina, I remember at that meeting, back when we were going to meetings in person, announced that about 150 petabytes of data had come off their sequencers that year. And as most people know, Illumina is one of the biggest, if not the biggest, genomic sequencing companies in the world. Now, if we compare that to other ways in society we generate data, so for example, for fun, and because it's public data, data that was generated that same year in YouTube, social media data, video content, it's about 50% bigger. Or if we compare it to other very large, international, robust research endeavors, for example, the work that's going on in CERN, in the Large Hadron Collider, it's about 6-fold more data than was generated that same year in that project. So it really is the case that genomics is generating some of our biggest databases of knowledge in the world, and is very likely to transform our lives over the next 5 to 10 to 20 years. We're already seeing this in a lot of the ways in which we use genomics in research. This is a figure from a paper from our next speaker, Dr. Alicia Martin, where she was looking at the growth in a particular database for genomic data called the NHGRI EMBOL GWAS catalog. This is one of a number of very large repositories for genomic data, and it really, this figure is showing that this really has grown exponentially, unsurprisingly, with what we know about how genomic technology has taken off. In particular, in the last couple of years, where cohorts and research endeavors are reaching the size of millions of humans per disease, per trait, hundreds of millions in aggregate across thousands of diseases and traits, because it turns out that this really was the tipping point we needed to get to in genomic research, where that data that's generated in research starts to become predictive for humans outside those research endeavors. And so we're really seeing a lot of how genomics is changing the way we do things in medicine. Since about 2008, we've routinely started to use targeted gene panels and whole exome sequencing for diagnostics for people in health systems who are suspected to have genetic disorders. We use genomics routinely in family planning, in various stages of family planning. We're starting to see genomics be used in other arenas. It's been used for a long time in pharmacogenomics. We're starting to see that come into more mainstream medicine. We're starting to see a lot of use of genomics in preventive health. So for genomic screening of putatively healthy individuals to predict risk, perhaps intervene early, perhaps even prevent morbidity or even mortality. And of course, this is happening in the context of a very data-driven, data-rich world around us, in which other types of biological information are really coming to the fore and being integrated with genomic data. We're starting to capture information in much more sophisticated, nuanced ways, including with nanotechnology, including with digital apps. And of course, you as a consumer have a lot of power to access your own genomic information. You, with a credit card, can go online to various different vendors and access genomic information to tell you about your health or your genetic ancestry. But with all these advances and opportunities, we have to recognize some of the challenges that we face. And again, I don't want to steal Alicia's thunder here. I'm sure she's going to show this in her talk as well. But really, it is the case that genomics is failing on diversity. And I'm not picking on this particular database. If you looked at any genomics database in genomic medicine or genomic research, you're going to see a very similar picture where the majority of participants, research participants in those databases, are representing European populations, more specifically Northwestern European populations, and many other populations around the world are underrepresented or indeed not represented at all. There's areas of the world with hundreds of millions of humans on the planet that we just know very little about genomic diversity in those populations. So the question is, recognizing this bias, what impact will this bias have on this future of using genomic information increasingly in medicine and society? Well, here's a study Alicia and I and several of our collaborators did a couple of years ago, where we looked at, we asked the question, if your databases are predominantly of European ancestry research participants, how will this impact using those databases to predict genetic component of disease or traits in non-European populations? And we showed that genetic prediction accuracy decays with the increasing divergence between the population that you use to generate the score you're using for prediction and the target population. And subsequently, many others now have made this observation, but it's nuanced and it depends on many different factors. So just thinking really about what are the key driving questions in an era of using genomics for health and medicine and wellness. Well, in my opinion, some of them are these. How do we really transform genetic research into medical knowledge? It's not a simple equation. It actually usually takes very many steps and a lot of science and implementation research to understand how genetic knowledge actually does translate into medical knowledge and where and when it's most appropriate to do so. And in doing that, I think we also have to really be cognizant and acknowledge the biases in our current knowledge bases. Who do these knowledge bases work best for or not? And not overpromise knowledge in populations for whom these databases are suboptimal. Once we've identified genes that are clinically relevant, how do we then systematically identify the individuals, the communities, or the populations who are at risk, and how do we engage with communities and understand other factors at play over a patient's life course that might impact that risk? And then another one that I won't spend too much time on today, but it's always it's something I think about a lot, is how do we integrate genomics broadly in health system for improving healthcare management? That is not a trivial task, and it requires a lot of thoughtful approaches toward provider adoption and patient adoption and health systems being incentivized to want to use this information. But let's go back to those first two questions, and I'm going to talk to you a little bit about work that we have been doing in the Icahn School of Medicine in New York City. And of course, New York City is notoriously a melting pot of global diversity. It has been a historical point of entry to the US for many hundreds of years. Recordings of people entering the US through Ellis Island originally, now touchdown at JFK. And it's a very, very diverse city. Here is one way of looking at diversity in New York City. This is taking a view of the 5 boroughs of New York, divided up into the 70 or so storied neighborhoods around New York. And this is using data from the 2010 census, US Census. And using that information, we can start to paint neighborhoods if they have a majority of a particular race ethnicity group as measured by that census instrument. And you can see in New York, there are many colors. So we have neighborhoods that are predominantly Asian, Black, Hispanic, or white. And then in purple, we have neighborhoods that are mixed race and ethnicity, no one majority population group. And understanding that, our institution, understanding the biases in our current research databases and how that maybe doesn't match the population that's walking in the door to our health system. Our health system is one of the biggest health systems in New York, serving 3 to 4 million New Yorkers. We made an institutional investment about 13 years ago in our own biobank to foster research that really represents the diversity of the citizens of New York. So today there's about 60,000 participants enrolled in the BioMe biobank. In general, if we think about race ethnicity groups, the proportions are about a third Hispanic Latino, about a quarter African American, about a third European American, and 10% other. Other can mean Asian American, it can mean mixed ancestry, it can mean these labels don't apply to me. And we see not a small amount of that, actually 4% of participants say that, which in a city of 8 million people is hundreds of thousands of people. So recognizing the limitation of simple race ethnicity categories, we also collect rich census level information, including questions like, where were you born, were your grandparents born? So taking a look at that data, we can straight away, just by asking people a simple question about where they were born, see that 65% of participants were born outside mainland US. So there's 3 or more grandparents born outside of mainland US. And if this is very consistent with what we know about immigration to New York City. The average age of participants in our biobank is about 55, 60. So if you go back those 2 generations, you're talking about world events that occurred at the beginning of the 20th century. So you see those patterns of immigration to the US in the distribution of where participants' recent ancestry is. And you know, overall, we're really looking at ancestry from about 160 countries around the world in the biobank in our health system in New York City. So in my lab, we study genetic ancestry a lot, and this is a very powerful tool to learn a lot more nuance about heritage and ancestry. This is principal component analysis, and to orient you here, I've also included reference populations from 7 different continents around the world: Africa, the Middle East, Europe, South Asia, Oceania, East Asia, and the Americas. And in gray here are the participants in the BioMe biobank from New York or New Yorkers. So the first thing I'm going to point out is that you can see that in terms of genetic ancestry, New Yorkers are overlaying with all 7 continents around the world. This is not surprising based on the figure I just showed you about where people say their ancestry is from. But the other thing I want to point out, at least in this type of analysis where we're looking at common variants shared across the global populations, there's no discreteness in population space. Rather, in the context of increasing diversity, what you start to see is a continuum of human genetic diversity that's really painting the picture of our common ancestry all the way back to sub-Saharan Africa. In my lab, we're also interested in thinking about ancestry that occurred in very, very recent timescales in the orders of the last couple of hundred to couple of thousand years, because that was a very interesting time in human population history. Our global population has exploded in time during that. We've become extremely adept at migration and dispersal and diaspora, and a lot of human factors have influenced, political and social factors have influenced our demography, causing events like bottlenecks, endogamy, migration, and admixture, which we think really impact the health landscape we see today in different populations across the world. So we use a method called detecting IBD, so identity haplotypes identical by descent, that arose within recent timescales, 5 to 50 generations ago, and by virtue of their linear decay, are still quite detectable at a population level. And we do this using methods, actually this is just coming out this week in Nature Communications, that are built to operate over massive scale databases. We just did this in the UK biobank of 500,000 people, for example, that are actually using some sort of machine learning approaches to really speed up how we can do this and enable us to actually do this fine scale analysis. So if we do this in the biobank in New York City, and then we operate a network community algorithm over that, we will see that there are groups of people who are robustly sharing much more IBD within the community than out with anybody else in the biobank. And then if we overlay information that we've collected in our survey instruments that include things like country of origin, race, ethnicity, or other questions about cultural grouping, for example, Ashkenazi, if you are Ashkenazi Jewish, then we see that many of these more nuanced population labels are highly correlated, and these communities are very good predictors of those labels in the biobank. So we see, for example, that we have a very good prediction of not only whether you're Hispanic Latino that we collect, but also if you might be Mexican, Dominican, or Puerto Rican in New York City. And this turns out to be very important because we know that there are diseases that track with demography, not only in shared genetic contribution to disease, but also the other components of environment, social, and behavioral determinants of disease. So we can do what we call an ancestry FIWAS. We'll come up with a catchier name soon, in which we compare by linking to medical records diseases that are enriched in communities or depleted in communities compared to all others. And sure enough, we can see, for example, in the Dominican community in New York City, we see a striking enrichment for many cardiometabolic medical billing codes, and captured here as fee codes, that indicate an enrichment of, in general, peripheral artery disease. And we don't see this enriched in other communities. And of course, we don't yet know if this is for genetic reasons, it's for health access reasons, it could be for health disparity reasons, but this gives us a very surgical tool to pay attention to this disease in this population. And as far as we've seen, there's virtually zero epidemiological literature about this, and so we are investigating it further. Furthermore, we can start to think about the impact of rich population structure on the ways in which we're using genomic data currently to predict risk. So here is looking at genomic prediction in European Americans in New York City. But what we've done is we've divided European Americans into those with Ashkenazi Jewish ancestry and those without. And you can see for all 5 scores here for common diseases like breast cancer and coronary artery disease, there is a difference, a mean shift in the distribution of those scores between those populations. And if you look at the bottom row, sometimes this can really impact a readout of a risk report depending what population background you are. So for inflammatory bowel disease, for example, the risk is much higher in Ashkenazi Jewish populations than in non-Jewish populations at the tail of the risk distribution at the high end. And then, of course, from a genetics perspective, what we're also very interested in is monogenic risk. We know that founder populations can sometimes be hidden in plain sight, particularly in cities. It does not need to be a force of cultural endogamy or isolation. Actually, the processes of diaspora and migration can lead to founder events and even forced migration. So looking at founder effects in the communities in New York, we see that there are many communities that exhibit signatures of bottlenecking, not only canonical founder populations like Ashkenazi Jewish or some South Asian populations, but also in Hispanic Latino populations. The timing of these founder events are very coincident with the colonization of the Caribbean and other parts of America. And those founder events can mean that genomic variants that are pathogenic for Mendelian diseases or diseases with high penetrance can be at different rates in different populations. So here we're showing that there is a 10-fold difference in your chance of harboring pathogenic variants, the BRCA1 or BRCA2 gene, between different communities in New York City. So at the highest end of risk are Ashkenazi Jewish populations, at the lowest end are populations from the Dominican Republic. But some other populations, for example, populations from Southeast Asia and certain Hispanic Latino populations from Central and South America, and not others from the Caribbean, are at high risk. And this is very important, nuanced information to know. And of course, it's also important not just at the population level, but also at the locus level, because we know, and we have shown this, and many others are showing this, not only in New York City, but also we've done this exercise now looking at 99 populations around the world, that you are much more likely to receive a report with an uncertain finding the greater proportion of African genetic ancestry you have in your genome, no matter how you self-identify or where in the world you live. And of course, we have to think about this in terms of genomic health and how we're actually bringing information into healthcare. Looking at it, this is a program that we have started. It's been running for 2 years now in the Institute for Genomic Health. We have a clinic for genomic health that sits on in clinical care and medicine. And here we're taking a genome first approach, but really tailoring this approach to diverse populations and at every step. So this includes not just thinking about what the CDC designates as tier 1 genomic conditions, which are gene disease pairs that have the best guidelines and practices for what you would do if you had this information in terms of actionability and treatment, whether the patient is symptomatic or not. We have actually expanded this program now to include another gene disease pair, Hereditary Ttr Amyloidosis. It's a TTR gene, and the reason we did this is because there's a common founder mutation in African ancestry populations, which in New York City is African American, African diaspora, and some Hispanic Latino populations, where the variant itself is above 1% frequency. So we have added this to our program and are currently performing research to actually screen participants for any of these genes and conditions, and then return results and intervene if necessary by referral to high-risk clinics or specialists, and then follow the outcomes in a diverse patient population here in New York City. So to conclude, and I'm just running a little over time, so I'll be very brief here. It's a very exciting time. We've got both a surge in genomic data. We've also got ubiquitous machine learning and artificial intelligence tools that are driving a lot of discovery, and it's leading to a paradigm shift in opportunities for translation and implementation. However, we know that access is not equitable, and we need to move toward more access and more equitability in these tools and resources. The intersect of genomics with medicine and society has really revitalized a debate about genetic ancestry, self-identity, and race. Inadvertently also, we have to be aware of race pseudoscience that's out there. And we really need to move away from solely relying on historical constructs of race in how we do science and how we practice medicine. Of course, genomics tells us about our susceptibility disorders, but it can also reveal the broader story about our demography, history, heritage, evolution, and that in itself can be correlated to social environment determinants of human phenotypes. So genomics not only can tell you about the heritable component of disease, but it can also be an excellent biomarker for environment, which can be used in health systems. And these questions, lastly, are likely to get more nuanced and complex. So we have a big need for translational research and implementation research in diverse populations and health systems. And with that, I'll just thank the folks who contributed to the work I showed, including Nora Abul-Hassan, Jillian Belbin, Sinead Kulaina, Ruhi Shiramani, Emily Soper, Sabrina Sukul. I showed work from your next speaker, Alicia Martin, as well. And a shameless plug, if anybody wants to do a postdoc in New York City. Thank you.
That's brilliant. Thank you, Professor Kenny, for a real powerhouse presentation. I should ask, are you able to stick around for the discussion session, or would it be better to allocate 5 minutes now for Q&A?
No, I am able to stick around. I'll have to step off and then I'll come back, but I'll be back for the discussion session. I might be 2 minutes late.
Okay, well, that would be brilliant. We definitely have a lot of questions for you, but thank you so much. We'll talk to you shortly. And with that, I will now transition to our second speaker that Professor Kenny kindly alluded to. I am delighted to welcome Dr. Alicia Martin. Alicia is an instructor in investigation at the Analytic and Translational Genetics Unit at Massachusetts General Hospital in the United States. She is an instructor in medicine at Harvard Medical School and an associate member of the Broad Institute. Her research examines the role of human history in shaping global genetic and phenotypic diversity. Given vast Eurocentric study biases, she quantifies the transferability of knowledge gained from large-scale genetic studies across globally diverse populations. She has published impactful work in this area, and I very much look forward to her presentation. Welcome, Alicia.
Thanks, Jason, so much for that kind introduction. I'm gonna go ahead and get my screen set up here. Hopefully showing the right ones. You're not seeing all of my emails.
Looks good.
Okay, thank you. Okay, so I'm gonna go ahead and get started. Thank you so much for the invitation. I'm really looking forward to talking with you all today about the really big critical importance of diversity in genetic studies. So Eimear already set a lot of this up very nicely in her talk, but as a geneticist thinking through an evolutionary and historical lens, I think we like to start at the beginning. In genetics, that means we need to start in Africa, where humans originated on the order of hundreds of thousands of years ago. As humans migrated out of Africa into the Middle East, into Eurasia, and subsequently into the Americas, they took a subset of genetic variation with them, which means that the average out of Africa population has about 1 million fewer genetic variants than the average African population. Importantly, there's more variation within than between populations. So when I say African population, this is the blurry boundary between other populations around the world. This human history then over the last 100,000 or so years informs a landscape of genetic variation across the world and is important to consider when we're designing our genetic studies. So it's the most exciting time I can possibly think of to be working in human genetics because our studies have just grown explosively over the past decade and a half or so. So the earliest genome-wide association studies are being conducted around 2006, which had hundreds to at very most a couple thousand individuals. And now we're routinely seeing genetic studies including millions of participants, which is just incredible. So we're learning a ton about human history, like I was mentioning, but also, of course, about the biomedical basis of disease from this exponential growth in our studies over the last decade. This I see is fundamentally really fueling the genetics mission, which is providing biological hypotheses from, importantly, unbiased associations that then fuel downstream analysis, including functional analysis, that eventually drive therapeutic insights and interventions. But of course, genetics has a huge diversity problem. So right now, about 80% of participants in our genome-wide association studies are of European descent, which is far out of step with the global population, where European ancestry populations make up about 16%. So this is a huge issue. If we look at the bottom part of this plot, you can also see that this hasn't really been on a trajectory to change so much. So our diversifying progress peaked around 2014 or so, and since then has stalled or perhaps even slid in the wrong direction for diversifying progress. Okay, so why does that matter? If we're fundamentally hoping to understand genetic variation and we expect that our effect sizes are consistent, well, then what's the issue? Well, we fundamentally need genetic variation is the ingredient to fuel our genetic discoveries. If we're putting blinders on and are looking at fewer genetic variants, we're then destined to find fewer genetic associations that are impacting a subset of the population and are not equitable across groups. So this is a huge diversity problem in genetics. But I think as we've looked at the social reckoning over the last year in particular, it's been so obvious how ingrained and systemic these issues have been. So I'm also just overlaying some titles from 3 of the big journals in science, Cell and Nature, where there's a reflection that this is not unique to genetics. Science has a racism problem. There's systemic racism, and science really needs to listen to these issues and to change if we have any hope of equitably serving diverse populations. So the outline of my talk today is going to include three main parts. So the first part is how critical diversity is for equitable translation of genetic studies, which I think Eimear also talked about, and other speakers in this session will be talking about as well. Then about how multi-ethnic and multi-ancestry studies are uniquely informative of genetic and environmental determinants of health. And then lastly, where do we go from here? How do we change the landscape and the picture of genetics right now to facilitate diverse population genetic studies? So a tool that I like to use to understand the genetic basis of disease and to understand the transferability and generalizability of our genetic studies is a polygenic score, which has been already discussed a little bit and is really being widely picked up in the media. So many of you have probably also heard of this. So polygenic scores use a genetic study that's been conducted before and uses that information to predict someone's genetic risk of having a particular trait or particular disease. So polygenic scores can be constructed on basically any phenotype of interest nowadays, with massive studies being pretty pervasively available. So you can use genetic studies to predict height, heart disease, cancer risk, and more social traits as well. So things like educational attainment have been widely studied with, some, I think, strong social caveats to consider. Looking at these distributions, these polygenic scores aren't super useful to individuals, but will likely be useful to populations as screening tools. So to understand a bit more why, we can look at the bottom part of this slide where you see the blue distribution making up those individuals who are healthy. These individuals tend to have a lower genetic risk of disease because they have fewer genetic risk variants. And in this red distribution, we see those with disease having higher genetic risk because they have more genetic risk variants. But you can see that there's a huge section of these distributions that's purple. So for the bulk of the population, we really can't distinguish too well those individuals who are healthy versus have the disease from their genetic information alone. So it's really in the tails of the distribution, those at the top 1% or those at the bottom 1% of these risk distributions, where we can say pretty well what's going on here from a genetics perspective alone. So people have talked about polygenic scores in a pretty wide variety of contexts so far. So one area that I have a lot of interest in and that we've been using polygenic scores in quite a bit these days is in research. So maybe it's not so useful that you predict whether or not someone has a diagnosis, for example, whether they have something like schizophrenia, because phenomenologically you can make that diagnosis without having polygenic scores as a biomarker. But when you have deeply phenotyped cohorts, you have things like readmission rates, you have things like what behavioral interventions have been tried, which sort of drugs patients respond to. These are areas where polygenic scores can serve as a useful research tool to try to understand the sort of trajectories of disease better. People have also talked about polygenic scores in clinical trials, where you can accelerate the efficiency of our clinical trials by using polygenic scores to identify those individuals at highest risk of disease. An area that people have talked about the most for polygenic scores, I think, is in routine clinical use. One area that I'm not so keen on is the idea of differential diagnosis. When we're trying to make that distinction between two related diseases, they tend to be highly genetically correlated, and therefore our genetic tools are not going to help us dissect the difference between common and complex diseases very well. But in preventative medicine contexts, this might be sort of an interesting and useful tool. So for example, in cardiovascular disease, we already use clinical scores that includes things like your hypertension, your BMI risk, your LDL cholesterol levels, and all these sorts of factors to stratify you into risk bins and to determine treatment strategies. Should you be on statins? Do you actually need to be on PCSK9 inhibitors? What should you do with your diet, your exercise, your lifestyle factors? So that's an area that has a lot of promise, particularly I think in cardiovascular disease and in some cancers like breast cancer, prostate cancer, and colorectal cancer. One area that's been talked about more recently, because new companies have been founded around this idea, is in the idea of embryo selection using polygenic scores. There's, I think, a lot of issues with this, and we need to have sort of a society-wide conversation about this. We'll have papers coming out in the next few weeks, sort of arguing about why this might be sort of a problematic or challenging idea. Okay, so with all of that as background, one core question that my group is interested in studying and trying to understand is, how do ancestry study biases in genetics impact the generalizability of the knowledge that we're gaining from these massive public investments? So there's a couple of important considerations to keep in mind here. The first, I think, is that biology is fundamentally shared across populations. And when we do our big genetic studies for all of these massive, complex disease studies, what we tend to find is that those variants that are really significantly associated with disease tend to have similar effects across populations. There are a few exceptions to that rule, but they're exceptions. So on the other hand, though, we talked about population history at the beginning of this. This influences the frequency of genetic variants. Of course, our environments differ across populations, and there are other complex factors that also differ across populations. So these differences in frequency, the differences of genomic structure, and these environmental differences mean that we need to have sort of a healthy dose of skepticism when we're looking at potential differences in polygenic score distributions across populations, and other big factors. So polygenic scores serve as a really exciting biomarker. I can't really think of a single lab test that can be so predictive across such a broad spectrum of disease. But what I see as the biggest challenge to implementation of genetics in precision medicine is this vast Eurocentric study bias. And the reason I say that is because when we look at polygenic score accuracy across populations, we see that we're predicting traits in European ancestry populations quite a bit more accurately than other populations. For example, about twice as accurately as in East Asian ancestry populations, and about 4 to 5 times as accurately as in African ancestry populations. So these are not small differences. I can't imagine running a lab test, for example, and getting back an LDL cholesterol marker that was that differentially accurate across populations. That would be sort of crazy, and I think would lead to a lot of public outcry, and justifiably so. Additionally, we understand the genetic basis of this. We know that our polygenic scores differ in accuracy across populations because they're fundamentally constructed from correlated variants, not necessarily causal effects, and because there are differences in allele frequency across populations and differences in LD across populations that date back to these population history maps that I showed earlier. And so we really need to diversify our genetic studies to have an improvement in this type of picture. Okay, so the next section of the talk is about how multi-ethnic studies are uniquely informative of genetic and environmental determinants of health. So to do this, I partnered up with a really talented grad student, Masa Kanai, who had worked with the Biobank Japan for quite a number of years. So the Biobank Japan is a hospital-based and hospital-ascertained cohort that has hundreds of thousands of individuals from Japan that all have contributed genetic and phenotypic data to a core central resource across different hospitals in Japan. The UK Biobank, on the other hand, is a population-based study. So we have compared these two studies, and our goal was to see if polygenic score accuracy in the UK Biobank and Biobank Japan gave symmetric and comparable accuracies when we're looking at different traits. So to understand this, we ran equal-sized GWAS for 17 different traits and 5 different diseases in these resources. And then we computed within and cross-population polygenic score accuracy. So what do we find? I'm not going to take you through all of the results because there's quite a number of them, but I'm just going to give you some of the key takeaways. So the first key takeaway is that our ancestry matched results are always performing better than the reverse of that. So for example, when we're trying to predict in Europeans, we do a better job when we have independent European discovery cohorts instead of different ancestry mismatched discovery cohort. But the largest meta-analysis is performing best. So combining data across Europeans, for example, and East Asians is giving us better results in both cohorts. The second key takeaway is that heritability differs by cohort and by trait. So Biobank Japan was really set up to study diseases that were ascertained in these cohorts. So there's really careful phenotype definitions based on the diseases here. UK Biobank was really set up to study continuous variation in the population. And consequently, the heritability of diseases is higher in Biobank Japan, and the heritability of continuous quantitative phenotypes is higher in the UK Biobank. So this means this phenotype precision is actually impacting our polygenic score accuracies. So heritability is actually not a fixed entity. It varies depending on how you're defining it and where you're studying it. So it's pretty context dependent. Okay, so where do we go from here? I see this as being really important. So I'm spending about half my talk on this piece because I'm just really excited about where we push forward. So I'm using this sort of as a roadmap for facilitating diverse population genetic studies to improve health equity for all. So I see there being really big needs that are sort of standing in the way of some major opportunities of diverse population genetic studies. So in the big needs category, I think we need better statistical methods that don't assume homogeneity and in fact relax to allow for greater heterogeneity in our studies. We need better data and community resources that make better use of data that already exists, moves beyond traditional boundaries of our studies, develops new resources, and grows to address gaps where data don't already exist. In hand with these data and community resource activities, I think we need to train the next generation of diverse researchers to lead in their home communities and in their home countries to improve research capacity that's diverse and will be interested in reflecting the diversity in which we hope to serve. And then with all of those in place, there are some huge opportunities that will stem from this. So we can identify new biology, novel associations, some low-hanging fruit that's sort of waiting because we've only been studying European ancestry populations by and large for the last decade and a half or so. Eimear already mentioned a few low-hanging fruit that's been discovered from her lab. Pretty cool. Fine mapping will also allow us to pinpoint causal variants because we can make use of this increased diversity in our genetic landscape to then more precisely identify which genetic variant is actually causing disease versus which is simply correlated and along for the ride. I've mentioned a lot about genetic prediction accuracy. This will improve for all, especially for underserved and underrepresented populations if we increase diversity in our studies. And then genetic versus environmental factors are not sort of mutually exclusive categories. We need to study these together and in concert to understand how these contribute sort of in an additive way to and an interactive way to impact health and disease. Okay, so diving into some of these different pieces, one area that we're really excited about is making better use of the data we already have. So if you're looking for a large discovery data set, we've recently released analysis from the Pan-UK Biobank project, which we started last year, and released GWAS results from the UK Biobank resource, one of the largest open biobanks that's available to share individual level data with researchers around the world. So this still consists largely of European ancestry populations, but it's a huge resource of about 500,000 people. So even if underrepresented populations make up only a few % of this, it's still over 20,000 individuals of primarily non-European ancestry populations, oftentimes the largest cohort that exists for many different phenotypes, given the vast breadth of phenotyping that's been done in the UK Biobank. So we've looked across ICD, electronic health records. We've aggregated these into sort of more interpretable fee codes that can be compared and shared across different healthcare systems. We've analyzed prescription data and done many analyses with this resource. So as of last summer, we released about 12 terabytes worth of GWAS summary statistics. That's a massive data resource consisting of over 16,000 GWAS of over 7,000 phenotypes across 6 continental ancestry groups. Many of the phenotypes that were collected as part of UK Biobank are somewhat culturally sensitive topics. They collected them. We ran GWAS for all of the phenotypes that we had at our disposal, so as not to elevate the importance of any particular phenotype more or less than others. But with that, we understood that there were many potential cultural sensitivities with this. So we developed frequently asked questions, or in this case, it was actually anticipated questions. So 17 pages of content targeted at lay and scientifically inclined audiences alike, describing the risks benefits and limitations, particularly in potential interpretation across populations for this resource. We used improved statistical methods. So we used generalized linear mixed models compared to previous analyses of UK Biobank populations. We analyzed many new phenotypes, which I already mentioned, and we are expecting to release some of our analyses from this resource later this year. So beyond that, we're also interested in collating data beyond existing boundaries. So with this Eurocentric study bias also comes a Eurocentric sort of collaboration that's sort of ingrained. And so we're trying to move beyond this and have started the Global Biobank Meta-Analysis Initiative. This now brings together more than 2.5 million genotyped samples spanning over 20 biobanks. So the power of biobanks to understand the genetic basis of understudied diseases is really powerful here. We're studying the genetic basis of over 14 different diseases that have traditionally not been widely studied. One example that we're spending more of our time on, because it's a larger study, is on asthma. There's incredibly wide prevalence differences reported here, on the order of like 2% versus 25% or something like that in the Global Biobank Meta-Analysis Initiative, which is sort of consistent with what epidemiology studies had identified before. But one thing that's really interesting is the remarkable consistency of genetic effects here. So we're diving deeply into that. We're also developing large-scale genomics resources. So this includes harmonizing and bringing together data from the Human Genome Diversity Project alongside the 1,000 Genomes Project, and making this available as a haplotype resource for future genetic studies. We're also bringing together many different genomes from continental African populations, which we're calling Genomes from Africa, or NOMAF, sort of a play off of NOMAD. I've listed sort of a link to some download resources in case you're interested in those. But this is building a resource of African allele frequencies and an imputation resource that will facilitate downstream African studies. Speaking of that, we're currently very actively working on addressing genomics gaps in Africa. So there's a big study called the Neuropsychiatric Genetics in African Populations Psychosis Study, or NeuroGap Psychosis. So we're on track to study the genetic basis of psychosis in over 35,000 Africans, I think closer to 40,000 Africans now. And we're doing sequencing at 4x depth rather than genotyping to build up genomics capacity and build up supporting genomic resources with this study as well as existing studies. This project wouldn't be possible without a big capacity building effort, which I'll come back to. And I just wanted to mention that we're really fortunate to be part of an umbrella project called the Populations Underrepresented in Mental Illness Association Studies, or PUMAs. We love our acronyms. So this encapsulates NeuroGap as well as several other projects involving colleagues at Harvard School of Public Health, SUNY Downstate, now Rutgers, and UCLA as the American PIs, and then their site PIs spanning African and Latin American research institutes, including in Ethiopia, Uganda, Kenya, South Africa, Nigeria, in Africa, and then Colombia, Brazil, and then several American sites consisting of Latin American populations. This study will be analyzing the genomes and phenotype data, including bipolar disorder and schizophrenia, in over 120,000 participants across Africa and Latin America. And then, of course, returning data to all of the sites and doing analysis in partnership with all of our collaborators. So we're really interested in understanding the genetic variants that contribute to highly heritable mental illnesses like schizophrenia and bipolar disorder, understanding the degree of consistency or inconsistency across different populations, and understanding how biology is perturbing genetic risk variants. Recognizing that I'm a little short on time, I'm just wanting to mention this last. So these projects would not be possible without a major investment in developing research capacity. So there's a program called the Global Initiative for Neuropsychiatric Genetics Education and Research, which we just shortened to GINGER, another acronym, apologies. So this is a multi-iteration program So the first iteration was a 3-year training program consisting of 17 research fellows across the NeuroGap sites that I mentioned. This consisted of workshops, online virtual weekly classrooms where participants learned about statistical genetics, population genetics, bioinformatics, psychiatric genetics and diagnosis, and phenotyping, epidemiology, all of that sort of thing. And then we tailored on-site trainings with curriculum jamborees focused on all of the areas that our collaborators and participants in this program wanted to learn about. So the first iteration of that program was a 3-year program. All of the Ginger Fellows graduated last year in 2020. The second iteration of this is a 5-year training program. So longer term, slightly fewer participants, I think 8 participants. These are 3 of the Ginger Fellows who I've been totally delighted to work with, Nastasha, Lorato, and Alan. They've looked at the generalizability of polygenic scores in diverse African populations. So just wrapping up here, polygenic scores are really exciting. I think they provide an objective biomarker that will help improve clinical models within our lifetime. But right now, they're really likely to increase health disparities due to vast Eurocentric genetic study biases. So to ensure that doesn't happen, we need diverse GWAS, new methods, resources, and research capacity, which we're very actively working on to address. And then we need to be cognizant of how culturally sensitive some of these topics are and communicate these responsibly and widely, anticipating the implications of our research. I'm happy to stop there. Sorry, I'm a little over on time, but happy to take questions if there is any.
Thank you, Alicia. That is fantastic work and a fascinating, fascinating talk. So is it best to take questions right now, or are you able to stick around for the discussion session?
I think I can, actually, maybe it's better to take them now if that's okay.
Yeah, no worries, no problem. So maybe I'll just start with one that comes to mind. So it seems that in order to address this Eurocentric bias that is so prevalent within the field of human genetics and is reflected in our databases, there seems to be 2 strategies, right? One is to try to make up for lost time and increase representation across the globe. The other is to think more creatively about making the most out of the data we do have and developing some statistical, sophisticated methods, which you're also on the forefront of. And it seems that at this point, it may be quite some time before representation in other parts of the world comes anywhere close to kind of the lead that Eurocentric data has kind of acquired. So to what extent do you think we should be investing in sophisticated statistical methods and developing that? And how important would methodology be in bridging this gap going forward?
It's a good question. I think our methods are only as good as our data. So as much as I'm excited about statistical methods and as active as I participate in like nerdy little conversations where we come up with a new and best method, really we're not going to be able to move the needle in a substantive way without improving the data resources that we need. So I think we really need to invest first and foremost in those big genomics resources and improving representation. I will say as a caveat to that we have noticed that there's disproportionate promise from these underrepresented populations when they are included. So for example, in some schizophrenia studies that we've done, we've seen that a study of 1/3 the sample size in East Asians, for example, compared to what we have in Europeans, already increases accuracy in East Asian populations that match that ancestry better than the largest European ancestry study. So there's a lot of ground to be made up sort of along the way. It's not going to be the case that we're only going to see promise when we have a biobank of 500,000 people in Africa, that we're going to see fair representation in Africa, for example.
Yeah, no, it's very, very interesting. So I guess going back to that Eurocentric bias as well. I mean, this is not new. This is something people have wrote about 12 years ago that the imbalance was there. And I think in the intervening 12, 15 years that bias has decreased only somewhat, and most of the increased representation is populations of East Asian ancestry. What's the problem? Why is it taking so long to get caught up and equalize representation?
Yeah, I think these issues are really systemic and really ingrained. So where are the samples available? They're available at the best funded institutions, which tend to serve European ancestry populations more. Where's the funding going? Well, it's going to the world-class researchers who have really established know-how, really established collaborations, which started earlier, and those earlier collaborations started, like, it's just sort of a trickle-down system that has sort of this systemic ingrained Eurocentricity to it. And so I think we're needing to actively push against that Eurocentricity and sort of prioritize resources towards diversifying our data sets and diversifying the investigators that are involved in these massive projects to ensure that we have the diverse makeup that we need to ensure genetics can benefit everyone equitably.
Yeah, brilliant. Well, in the interest of time, we should probably move on to the next speaker, but thank you so much for a very incredible, interesting talk. It's always a pleasure to hear the work that you're doing. So thank you very, very much. Cheers. And with that, I'll now transition to our third speaker for the event. I'm very pleased to welcome Professor Aimé Lumaka. Aimé is Associate Professor of Genetics and Pediatrics at the University of Kinshasa in the Democratic Republic of the Congo, and the coordinator of a genomic medicine platform at the University of Liège in Belgium. His current focus is on the clinical and genomic characterization of rare diseases, including in African patients and severely ill children in Belgium. He is also the chair of the H3 Africa Rare Diseases Working Group and is leading the African Rare Diseases Initiative. It is my great pleasure to welcome Professor Aimé Lumaka.
Thank you very much, Jason, and thank you to the organizers for this interesting topic and for the panel that we have been able to put together. So today I'm going to talk to you about the approach to improving healthcare for rare disease patients in the world by doing our research in Africa and on African participants with or without rare diseases. My disclosures. So regarding the context, a rare disease is a disease that affects 1 in 2,000 people, according to the definition from the European Union. And this is a heterogeneous group of diseases. It's not one disease, but it's much to be seen as a group of diseases that have very low frequency. And about 80% of those diseases have genetic bases that have been discovered, meaning that for the remaining, the genetic basis, it's yet to be uncovered, and there are also other diseases that are not known yet. Although these are individually rare, but if you put them together, these are much more frequent events. And it is estimated that between 3.5 to about 6% of the world population could be affected by one or in another rare diseases. So if we try to put this into perspective, this would make up between 42 to 71 million living people in Africa alone, which is a very big community of individuals and family that are in need for diagnosis and care as well. So if we look at the distribution of those diseases, what we see based on existing publications that they are different, they don't have the same frequencies. Some are much more prevalent than others. And what is interesting is that the more disease has been studied, the more it tends to be reported to have a high frequency, which is in a way hiding a bias that could do that some diseases that are receiving very less attention because they are not of interest for the population where the funding is being offered might be ignored. Actually, we might not have the very accurate figures for those diseases because they are not prevalent in the wealthy communities where the funding are being given or provided. And if we compare across the globe, we will see that some diseases are much more prevalent in Africa, such as the sickle cell anemia, which is the most prevalent monogenic diseases known thus far, as compared to cystic fibrosis, which a few decades ago was reported to be absent from the African population. So the rare diseases in itself, the definition itself and the distribution carry the seed for health disparity. But across the decades, as Jason mentioned at the beginning, multiple advances has been achieved. The major one was the completion of the Human Genome Project, which has provided us with the consensus templates of DNA to which we could compare one's DNA to identify changes that might be of interest for the health condition of that particular individual. From the fact that we were able to have a reference genome, this has really given a leap forward to big tech companies in terms of developing technology that would allow us to access someone's genome to read the genetic composition of one particular individual by using rather short reads or long reads, or even now that we are able to use technologies that can fit in our pockets, like a cell phone size sequencing technologies that we can actually use on the field to read the sequencing or the sequence of one particular individual or one particular infectious diseases species. As we are now able to read much more in depth and much more widely, we are able to read the genome at the entire genome level, the DNA level. And this is the whole genome sequencing, a very wide sequencing technology that allows us to identify much more variations in someone's DNA. Here you see the screenshots of the two patients with rare diseases, and if you look at the single nucleotide variant, you would see that they have more than 8.3 million variations in their genomes. So this is one of the advances that we have been able to achieve to the fact that we have the reference genome that has allowed sequencing technologies to evolve much more quickly, allowing us to explore the entire genome much more wide and much more deeply. So this has been taken advantage by pharmaceutical companies due to the fact that we are able to identify much more genes that are causing rare diseases, and it has helped to accelerate the identification of orphan drugs, those drugs that are important for care or treatment of those rare diseases. So the pace of discovery of genes is correlated to the pace of discovery of orphan drugs. So now we are able to quickly sequence someone's genome, and this can be achieved in less than a day, because very recently, a couple of weeks ago, the Rady Children's Hospital reported that they have been able to sequence the entire genome of patients in 16 hours. And you can see multiple efforts, similar efforts are being undertaken in Australia, in UK, in USA. And this is what we are doing also in my lab here in Liège. We are also running a rapid whole genome sequencing for critically ill patients, where we are able actually to return results in 40 hours and allow clinicians to integrate the result in the treatment. So while those progress are being recorded, while we are able to sequence more, while we are able to use those informations to improve the care we can provide to patients, the same progress we are really proud of are also creating disparities between populations from one part of the world to another, from one level of development to another, a lower level of development, especially in Africa. The best illustration is the distribution of sequencing technologies. As we could remember, at the end of last year, one of the biggest tech company, Illumina, reported that they have been able to solve the 1,000th high throughput sequencing machine. So this allowed them to reduce the cost so that they could be able to offer a whole genome sequencing at $600 US. interestingly, if we look at how those instruments are distributed across the world, I try to search and I try to contact people to make sure that I didn't miss something. What I noticed that's the only instrument that I can actually confirm that the African continent has one NovaSeq is one that is the 54 gene in Nigeria, which means that these technology advantages are not available to some part of the world. And this reflecting the number of sequencing or genomic data that are available. It is estimated that among all the data that are available for the moment, only 3% are from African individuals. So that is the technological disparity. The second one is data disparity. Yeah, it's very important to notice that all those technologies that are allowing us to sequence more, to get much more information, are only the first step in the genomic medicine, because in addition to accessing genomic data, we also need to be able to properly interpret those informations with regard to our patient's health condition, with regard to the phenotype the patient is presenting. So without the interpretation, those data are not really helpful for the community or for the patient themselves. And so this is one of the challenges we need to address, to be able to diagnose and to, when it's available, provide appropriate treatments. And for families, they are also in need for counseling. They need to know what is the recurrence risk for the family, and whether there is or there isn't a targeted treatment that they could benefit from. So what we do on a daily basis in the rare disease field is, when a child or a patient come to the consultation, a clinical evaluation is conducted to try to capture the phenotype of these patients. And before the era of sequencing first, this phenotype was used as a filter to decide which genetic testing would be prescribed. And then the sample will be collected, sent to the lab. Once the sequencing is performed and the quality of sequencing is checked, then those data would go through the interpretation process. The interpretation of processes two major steps that require available information from the literature, from databases. One of those is whether the variant that have been identified in the patient are known to be present in population database, what we call reference databases, those databases that are supposed to be deprived from individuals with rare diseases and their relatives. We also need to know whether those variants that we have identified in our patients are or are not already reported in patients with similar phenotype. So those are very important sources of information that we need to have in order to conduct a proper interpretation. So good phenotyping, good information in databases, should it be reference population database or disease-specific databases. We need information to be available there. So what we are encountering on a daily basis is that the phenotype or the phenotypic description and the spectrum of some well-known diseases, genetic diseases, are still not well characterized in African individuals. A few years ago, we conducted this study where we tried to compare Down syndrome patients from Congo to Down syndrome patients from Belgium. We conducted the clinical part where we provided photographs of those patients to establish clinicians in Europe and in Africa. We really saw a big differences in inter-rated difference that was significant. And we also did this using machine learning. We created artificial faces that we compared using face to gene. As you could see, there was almost no resemblance in the gestalt, this face that was created between the African patient and the Belgian patient, irrespectively to the fact that they had the same underlying genetic abnormality. So there is a need to conduct the proper characterization of the phenotype in patient in order to improve the clinical diagnostic yield in African individuals. And we have taken some steps by trying to characterize the reference population, the reference newborn populations in Congo, to allow people that are conducting clinical evaluation to be able to exclude features that would in other areas be considered among dysmorphic features, but that are rather common features for the Congolese population. And this study has been recently published in the American Journal of Human Genetics, where we provided some frequencies of the features in our community and made some comparison to other communities in South Africa and Asia. Another problem is that we are identifying, as my previous speakers, co-speakers said before, we are identifying a huge number of variants which we don't know what is the medical or the clinical significance of. And these studies are proving that if you sequence an individual of African origin, you are much more likely to identify a huge number of variations. And those variations, you don't have information in the reference databases to be able to provide a proper classification for those variants. And this study of 15 hunter-gatherers identified more than 3 million novel variants. A recent study published this year in Nature using the H3 Africa data identified more than, a little bit more than 3.4 new variants that are not in any known database. Here we compared some of our Congolese patients who benefited from whole exome sequencing to some Caucasian patients that have been sequenced through the same technology. What we noticed that the proportion of known variant was much more higher in Caucasian patients as compared to Congolese patients, which makes it difficult to reach the interpretation of variants in patients of African origin. And this is understandable if you look at the most reputable source of reference frequency, which is actually NOMAD. You would see that the proportion of individual of African or African American is increasing, but still very small. And especially if we break this down into proper African data, it's only limited amount. And the same study that I mentioned using the H3 Africa also reported that although the global number is increasing, but we still have multiple gaps where we don't have data actually, where we would need to collect data to capture the fine granularity of the African genotype, given the high diversity of genetic data in Africa, which is a well-known phenomenon. So if we go to databases of disease variations, we see the same thing, that is the, if you look at the ClinVar, you would see that submissions from African countries are very, very rare, which means that even when we are sequencing African patients, we are not reporting these informations in the public databases to allow other people to use the same information to provide a proper diagnosis services to their patients. And we also conducted a study that showed that the yield in the intellectual disability patients in Africa is very low, even though we use a combination of multiple genetic testing approach, the yield is still very low. An illustration of that is this publication that we recently made in the American Journal, where we published these patients. It took us 6 years of analyzing and reanalyzing the patient until we find a record, a recent record in ClinVar that allowed us to reach the conclusion. So the data disparity is actually leading to the health disparity. many African patients are still undiagnosed, even though they have genetic data, which means that multiple rare diseases, they remain undiagnosed or uncharacterized. So the care also impacted. This publication looked at the existing policy or strategies for supplying orphan drug to different communities. There is almost nothing that is existing in Africa. So we are trying now to address this disparity, but we need to recognize that this is not something that is specific to Africa. Those publications that I have compiled are showing that the fact that data informations are missing in databases are leading to false diagnosis. This is from USA, this is from UK, this is from I think this is from Lebanon, and this one is from a Middle East country as well. So recently we received this email from a colleague from Canada. He sequenced patients of African origin and identified 3 different variants he couldn't interpret. And he reached out to us. We tried to look at all our African data to see whether we could help. And we provided the frequency of those variants in African individuals so that he could be able to properly interpret the frequency in his patients. We have also been contacted by people from USA that are working in area where you have huge population of African origin. They are also facing the same problems. A huge number of variants they are identifying, they are not able to reach a proper classification. So what we are doing with the H3 Africa, which is a project that is funded by the Wellcome Trust and the NIH, we are aiming to develop a sustainable and collaborative African genomic research network that is intended to improve health. There are multiple objectives to this, and especially for the rare diseases, we are trying to facilitate collaboration, share experience, share resources, and try to insert our views in all the policies that are being developed. And we are working in collaboration with ethnic groups and phenotype harmonization. For the future, we are now going to toward the African Rare Disease Initiative, which is specifically willing to address data disparity using multiple mechanisms, which includes development of or increasing capacity for generating data, sharing those data through beacons, and creating a type of medical record that could be used by everybody and would facilitate data sharing. We are working in close collaboration with the CleanGen that is really helping, and we are willing to engage with much more people, as we are already doing with those researchers that are outside of Africa and are working on patients or community that have ancestries from Africa. So this means that there is a group, or there are groups that are missing. We don't have collaborations with people from UK. We know that there are a big community of people with African ancestry in UK. We'll be very happy to collaborate with you and see how we can help each other, so that ultimately we can help patients with rare diseases through those type of collaborations and researches. So I would like to thank you for your attention, and these are my support: University of Liège, University of Kinshasa, the H3 Africa, the DDD Africa, which is a replicate of the DDD UK, and the H3 Africa is funded by the NIH Wellcome Trust and is actually supported by African Academy of Science. As I mentioned, our African Rare Disease Initiative project first phase has been submitted to the NIH. We are waiting for the review, which is ongoing. And a great thank you to Dr. Jerry Mubungu, because she conducted many of the researches, which I include the results in this presentation. Thank you very much. Jason, back to you.
Thank you so much, Aimé. This is clearly important work and just fascinating research that you're involved with. Are you able to stick around for the discussion?
Yes.
Perfect. All right. Well, in that case, we should probably move on to our fourth and final speaker, but don't worry, we'll get back to you, Aimé. With that, I am absolutely delighted to welcome our final speaker, Professor Keolu Fox. Keolu is an assistant professor at the University of California in San Diego. I understand, Keolu, you're calling, it's rather early where you are. I think it's just 7 a.m. if I'm not mistaken. So we are very grateful that you're willing to join us so early. And Keolu is, as I said, he's based at the University of California, San Diego, affiliated with the Department of Anthropology, the Global Health Program, the Hallijolu Data Science Institute, and the Climate Action Lab, and the Design Lab. And the Indigenous Futures Institute. So very busy, Keolu. Your work focuses on the connection between raw data and a resource, and the emerging value of genomic data health from indigenous communities. Keolu has experience designing and engineering genome sequencing and editing technologies, and a decade of grassroots experience working with indigenous partners to advance precision medicine. Interestingly, Keolu is also the first native Hawaiian to receive a PhD in genome sciences. And as always, I'm very much looking forward to Keolu's presentation. Welcome.
Thank you, Dr. Torres. Is there a way I can turn my camera on? It says that I'm unable to start my video.
Should be able to start video. Kath, does Keolu have?
We're on it.
Oh,
good morning, everyone.
Hello.
I appreciate it. Thank you so much for the kind introduction. Thank you so much for inviting me. It's lovely to see so many friends and productive scholars thinking about health disparity, genomics, and including a more diverse and broad group of people in the future of human genome sequencing and technology development. So awesome to be here. And I really appreciated the art that you guys created to accompany this conference. It's really beautiful and thought-provoking. So well done on that. So I guess I will begin my screen share. I have a lot to cover. So yes. All right. So thank you so much for having me. As Dr. Torres said, I am here in lovely Kumeyaay Nation, or La Jolla, California. And I have a number of affiliations. I am a co-founder also of NBDC, the Native Biodata Consortia, and I'm really proud to say that we just started the Indigenous Futures Institute here at the University of California, San Diego. I'm originally from Pololu Valley on the Big Island of Hawaii. It's a beautiful place if you've never been there. It is one of my most favorite places in the world. It's also the same exact geography that shaped my ancestors' genomes and probably contributed to many of the health disparities that we have today. Currently at UC San Diego, it is the home and the epicenter of much of the genome sequencing technology that many people around the world use. It's a thriving ecosystem for biological innovation in both medicine and predictive and preventative disease. And it's also perennially ranked the #1 institution in the world for surfing as a university. So we're really proud of that by Surfer Magazine. And here's me, another day in the office here. Lovely place to work and have a work-life balance that informs the trajectory of our science. Shameless plug here. I won't talk about this work. Brand new paper. I believe it is actually from Oxford University Press, and it's published in MBE. And the title of the paper is Abiogenetic Variation in Neanderthals and Denisovans with Fernando and Emilia. Feel free to check this brand new paper out. Pretty fun stuff, imputing haplotype structure and thinking about abio variation and integration in archaic genome assemblies. It's probably about 4 to 5 years of work, so it's nice to see it hit the press. Today's talk, though, I'm going to focus on 3 projects and ideas that I'm fortunate to work on. One is an exhibition and a museum setting, very different. And and it focuses on the history of eugenics and scientific racism in Hawaii. The second is functionally investigating private genetic mutations using base editing tools, if you're familiar, or prime editing tools. And the third is building more equitable systems for the management of health data for Indigenous communities and moving towards what we like to call Indigenous data sovereignty and technological independence and democratization of many different tools, including algorithm fairness, et cetera. So first, let's begin with something that I was privileged to work on with many different partners at the Bishop Museum in Honolulu. Beautiful place if you've ever been there. Our exhibit, Regenerations, Challenging Scientific Racism in Hawaii, is going to be on display until this October. So if any of you have the opportunity to make it to our beautiful islands before then, please write me an email and I will try to get you accommodations in terms of your ticket. I got to work with these lovely ladies, curators and archaeologists and anthropologists, and it was an auspicious day when we had our opening. You can see this anuenue or rainbow in the background. And what we did with this exhibit, Regenerations Challenging Scientific Racism in Hawaii, is we focused on some central ideas around the history of eugenics in Hawaii. And we found a catalog of medical images of our community members, many of which have living descendants in the Hawaiian community today. And we utilize that position in those images, those photographs in an archive, and we built and co-designed an exhibit around those images with the Hawaiian community, many representatives from different islands in our archipelago. Here are some of those images on live display. This is my friend Nalani with her newest baby, and they're looking at a picture of themselves and their family and their ancestors. And it's a lot to take in, and it's a tremendous educational opportunity so that our community members understand how and in what way archival and medical data is generated, and what is the history of exploitation and extraction in Indigenous communities. In a lot of ways, the anthropology of the past is not that different from the anthropology of the present. We have this explosion of interest in ancient genomics and the generation of data. Much of this coming out of labs like David Reich's lab, for example, does not feature community-based consent. It does not feature consensus building with the communities in which much of this data is extracted. And dare I say it, there are many different forms of human rights violations that are happening. And so we talk a lot about that idea. And it's not just me who feels this way. It was, featured in the New York Times and many, many, many other things, and creating this tension within ancient genomics and population genomics broadly. So much so that more ancient genomes have been sequenced since 2019 than in the entirety of history. This is a feature and piece that I wrote with John Hawks in Nature last year, and you can just see that sharp, incline and exponential growth. Coupling that with the number of ancient DNA laboratory workflows that have been constructed since 2010, you can see there's an intense interest in generating this type of data. And we know that this many laboratories and this many new projects and the scale and the anxiety for discovery generally does not feature the communities of people in which these genomes have been derived. To make matters even more complex, institutions like Christie's and Sotheby's and many other online auctioning platforms and big data sort of ecosystems like Instagram are thriving ecosystems where people can pay for and purchase Indigenous people's remains online for the highest bid, if you will, so to speak. So we featured that concept or idea throughout our exhibit as well. Here's a peek at part of our exhibit. We feature many artifacts that contain our king's hair, our ali'i hair, genealogical connections to our monarchy, knowing that these artifacts do contain DNA. And it's our job to protect them and ensure that others don't begin to extract genetic information from these ancestors. Because they fit very neatly into the big data economy and apparatus. For example, in 2018, 23andMe sold access to their database for $300 million to GlaxoSmithKline, and the value of genomic information and data broadly is on the rise. In 2020, we see the Blackstone Group acquiring Ancestry.com for $4.7 billion. This is just an indication that DNA sequence information and data broadly is worth a tremendous amount of money and has the potential for all kinds of things, prediction and modeling in certain contexts. We feature many of these new and emerging ideas and technologies throughout our exhibit as well. We have an entire DNA lab in your backpack, many mobile and point of care features and technologies. We also created a kind of fake company called the Biocolonialism Trust, where we talk about ideas around consent and many other things related to the consensus building and consent process. None of this would be possible without the living descendants of those individuals today. It's always a true pleasure when you get to co-design projects and art and science and recombine them into an opportunity for education for our community. So it's just been beautiful. It's got critical acclaim. We never thought that our little exhibit at our little museum would be featured in the New York Times and every other newspaper and magazine. It's tremendous. For those who funded this project, MIT, Emerson Collective, and many other folks, Hawaiian Airlines, thank you so much. Next, I'm going to pivot and move more towards mechanistic ideas and challenging and thinking about empowering Indigenous communities and rewriting history using genome editing tools. And you're probably wondering, what is this guy talking about? So many of you know this, and I'm sure Alicia Martin and others have kind of tuned you up on the fact that we have a diversity issue in the individuals that are included in the future of predictive and preventative medicine. Famously, a paper from Carlos Bustamante, Esteban Bouchard, and others featured this in Nature called Genomics for the World, where they showed that 96% of genome-wide association studies exclusively feature individuals of European ancestry. Moving on to today, we can look on Melinda Mills' GWAS Diversity Finder, and you can see that number is kind of steady around 90%. We have a number of initiatives that are trying to increase diversity broadly. We just heard about H3 Africa. We've heard about the All of Us program to sequence 1 million individuals in America. But one very dangerous part about all of this research that we're not talking about is the fact that 72% of genetic discoveries have taken place in 3 countries, meaning we're privileging a certain way of interpreting that data and the many ways in which we instill and bake in bias into all of the projects that we create using human genome sequence information. And that leads to what Stephen Jay Gould and Richard Lewington referred to as just so stories. I'm sure many of you are familiar with this idea. These are tall tales about maybe taking something that's a correlative at best speculation and having no causative evidence about what's going on here, right? And this is based off of a child's book from Roger Kipling, who wrote Kim and The Jungle Book and many other kind of stories. And we can apply that to population genetics, and we can look at many of these different stories. Here's a map from Sarah Tishkoff from 2015 that I really like, looking at the global distribution of locally adaptive traits. And some of these ways that we interpret this type of correlative data is correlative at best. Sometimes it's accompanied by functional data. Sometimes they're A+ stories. For example, sickle cell anemia, malaria, and HBV. That's a really solid story where we have mechanistic evidence. This has been reverse engineered in cell lines using CRISPR-Cas9 and so on. We have other stories that are a little better. EPAS1 in high elevation populations being involved mechanistically in the HIF pathway. But some of these stories don't actually stack up, and the narratives in which they are rooted are not really in line with the biology, and they discredit a lot of the accomplishments of people. And namely, they do not really think about the effects of colonialism as a variable on the genome, for example. One of them that I've highlighted because it's identified in Samoa initially in 2015 is a genetic mutation in CREBRF, first found in Samoa. Again, It is supposedly a thrifty genetic mutation, which is a really archaic biological anthropology kind of theory and idea. It's downright offensive and racist to a lot of people. And it's interesting that this mutation is discovered. And if you know anything about the endocrine system and metabolic disease broadly, you wouldn't expect to find a mutation that predisposes to obesity, but protects against type 2 diabetes, which from the therapeutic point of view is very interesting. So let's dig a little deeper into this mutation. Again, entitled as a thrifty mutation discovered in Polynesia. Obviously, I'm from Hawaii, so we take issue with that. But the media, as usual, loves to run away with these things. You have the Japan Times saying that this is the gene that may benefit sumo giants and how this powerful obesity gene helped Samoans conquer the Pacific. Now, the idea here is that this mutation allowed our ancestors to discover all of the far-flung island archipelagos in the Pacific, including Hawaii, Aotearoa, Tahiti, and Rapa Nui. That's just not true. Being from the culture, I can tell you that we're the greatest voyaging and navigating community in history. We utilize complex systems of science and the scientific method, including and not limited to ocean, swells, winds, currents. And at night, we got really, really busy, and we utilized the celestial sphere to find and identify some of the most isolated places on planet Earth. At no time did we ever have problems with calories or provisions on the boats that we were on. Actually, in the Disney movie Moana, they actually had a pig and a chicken on the boat. We had some very advanced horticulture. And there's even evidence from yesterday, in fact, showing, this is something we've known, sorry, Shackleton, that our ancestors reached Antarctica at least 1,000 years ago. We saw recent research from Andres Moreno and Alex showing that you can actually confirm this, that our ancestors made it to South America as well. So again, these types of narratives pop up. They invoke the thrifty gene. They invoke these reasons why indigenous people do have some of the highest rates of metabolic disease in the world. And when you say that it has to do with innateness and not that it has to do with the consequences of colonialism and the consequences of having a lot of our land stolen and taken away illegally and not having access to reefs where we like to fish, hunting rights, food deserts, and a lot of the issues that come along with that, it lends to and creates these oversimplified narratives about why common complex disease exists in certain communities. This mutation isn't just found in Samoa, it was found in many other Polynesian communities, including the Maori from Cook Islands, Tonga, Hawaii. It was even found in the Chamorro from the Marianas Islands, who are supposedly Micronesian, challenging the boundaries and the ideas and the fluidity in which we've created these divisions between island communities. What we like to do in this situation is take that exact mutation and utilize base editing technology and reverse engineer this mutation in the appropriate physiological cell line. And this is a really cool prospect because it allows us to, and here's the confirmation of that. This is recent work and some Sanger sequencing data. I'd also like to point out that we did this for a fraction of the price that they'll charge you at a company like Synthago, maybe 20 to 25 grand. And we can then actually assess whether this is a thrifty mutation. We can test for insulin sensitivity, inflammation, metabolomics, etc. And it's highly adaptable for other genes. We're doing this with archaic specific mutations as well. So it's a very fun time to be in reverse genetics. Just wanted to showcase a little of that. But one of the issues that's arising around many of these mutations is that non-Polynesian scientists have filed a potential patent claim around this information. You're probably asking yourself, Keolu, what are you talking about? What happened with the Mary Claire King versus Myriad Genomics court case? How did this happen? How are people attempting to commercialize the exclusive use and physiological response of this allele as broadly as possible? Now, this patent claim didn't necessarily work out, so to speak, but the fact that individuals would want to do this and commodify this type of genetic mutation without including members of our community is downright horrendous. And so because of that, motivated by that and many other things, I'll finish by talking about building more equitable health systems. It seems like every week another paper comes out talking about genomic insights into population history, for example, and biological adaptation in Oceania. This paper is by Luis Quintanilla Mercy. And you can see that they have a lot of problems on the ethical end of things. The study reveals an exciting history of humans in the Pacific, but critics, meaning community members and our representatives, blast the lack of local indigenous input. And this happens every week, that there's this disconnect between what scientists believe as engagement and what a true partnership really is. So it seems like many times we have scientists that are seeking authorization and not true consensus building and connectivity. And this is quite ubiquitous throughout our field. And that leads us to indigenous data sovereignty. When we put the community in charge, when we put the community in the driver's seat, when we put members of our community and we educate them and we are allowed to make decisions and interpret data about this, things operate a little differently. As many of you know, data is power. Data as a resource has surpassed oil as the #1 commodity on planet Earth in 2018, and its value is only on the rise. Genome sequence data derived from human beings, whether it's our complete human sequence or our microbiome or many other things, fits very neatly into that big data capitalism ecosystem. So we would be foolish as community members not to recognize that and create systems of technological independence around it. We put out a review on this, check it out, it's in Nature Genetics. It kind of puts out the playbook for many different ideas, including recognizing indigenous rights, opportunities to share and think about privacy and security, and also the potential for commercialization and partnership agreements and commodification with indigenous partners. Regeneron knows this. They're very interested in identifying many genetic mutations that are sort of the gift of our migratory and diaspora and history, and fast tracking those to develop new forms of pharmaceutical drugs. Look no further than PESC9 inhibitors as a very successful opportunity for this to happen. This is just the beginning of many different paths towards drug development. And if you're me and you're from Hawaii, you definitely want to see your community get as large of a piece of the pie as possible, whether it involves intellectual property, and many, many opportunities for royalties or subsidized or free access to drugs that were derived from information that was extracted from our communities. We want to avoid the development of $300,000 drugs like Vertex's pharmaceutical drug, or if many of you are paying attention, there's another Alzheimer's drug that hit the market for $58,000 a year. And I know what you're saying. If you're in England, you got you have universal health care, but this is a very large problem in terms of drug development. And I talked about many of these different ideas and the validity of approaches for equitable benefit sharing in the New England Journal of Medicine. We talked about the data flow, collection, storage aspects of things, the current research and development model where you have government subsidized grants and information that's derived from taxpayers' dollars being driven and taken over the goal line by big pharma, highly problematic, and not situating and empowering individuals who have provided graciously access to that genomic information. So I kind of contrast two different models for collective interest or community trust model and individual interest and fractional ownership. Feel free to check that paper out. And this has allowed me to create and think about and partner in the development of many different types of policy, both on the domestic and the global level, thinking about and recognizing data as a resource through the World Economic Forum, the UN, and many other institutions. This is just the beginning of juxtaposing this top-down and bottom-up approach to thinking about empowering people. Finally, I will end by just briefly talking about the future of Indigenous genomics. We're deeply thinking about vertical integration, like any good capitalist. We have borrowed ideas from folks like Henry Ford and Amazon.com, the largest company on planet Earth. In many different ways, we have been doing this. As a founder of NBDC, we're doing research for natives by natives, and we are vertically integrating every single piece of technology to put our communities in the driver's seats of their information. And these are many of my partners and board members for this initiative. We're thinking about enhancing ethical research with Indigenous communities and putting out the playbook for that. There's no substitute for grassroots research. We are democratizing technology through a number of different educational initiatives. This is my friend Hayden Henry doing his first PCR in Aotearoa. And we had our first Sing Indigenous Genomics Conference in 2020. This was a first international Indigenous Genomics Conference with all Indigenous speakers, graciously sponsored by Illumina and Nature Genetics. And we are having Indigidata, and this is coming up next week, I believe, and we are empowering the next generation of Indigenous data scientists and arming them with tools to think about many of these issues. We are remapping the human genome, and this was a really cool paper from one of my PhD advisors, Evan Eichler, thinking about creating population-specific genome references and the idea that they identified new human genes, and this is a huge, exciting new avenue for development. And finally, we've been Indigenous futurists for a long time. Here in Hawaii, we created some of the first color newspapers in the world. We had electricity in Iolani Palace well before the White House, and we will always use the most cutting edge technologies to empower our communities. And that's what we're doing at the Indigenous Futures Institute. We're not just thinking about health and the future of hospitals and the health of future of healthcare. We're thinking about energy sovereignty, land rights, and many, many other things. And thank you so much for this opportunity. I hope I didn't go too far over.
No worries, Kalu. That was fantastic. Thank you for that. Clearly thought-provoking and important work. And at this point, we'll transition to the discussion session. So if the other speakers could turn on your cameras if you're still around. But before we maybe start the discussion, there's something I really want to ask you, Kalu, and you touch on this a bit, this concept of Indigenous data sovereignty, which at its heart involves granting more control of genetic information to community stakeholders, tribal leaders from those communities. When it comes to decisions regarding data access, data usage, benefit sharing. And I was reading, I read your piece, your commentary in New England Journal of Medicine, and I also read a recent paper from Crystal Sosi in the American Journal of Bioethics. And she also touches on this concept of Indigenous data sharing and talks about the fact that in terms of setting the terms of data access, data usage, this is often established, by research funders and research institutions that typically doesn't involve the consultation of community stakeholders. And usually when Indigenous communities are invited to participate in genetic studies, it's usually either in such a way where they have to accept terms that have been preset that aren't necessarily aligned with Indigenous data sovereignty, or they are excluded from the studies altogether and risk missing out on valuable biomedical insights. So what do you think should be the path going forward? And I'm thinking in terms of somebody like myself who certainly wants to be inclusive in terms of studying data from people all over the world and leveraging the information from different populations, but don't want to do this in a exploitative way.
Yeah, thank you, Dr. Torres. Great question. A lot to unpack there. Crystal is a dear friend to me, and man, brilliant perspectives from her always. I think maybe philosophically we have to zoom out and ask questions about what the greater good is and whose greater good we're talking about here when we establish networks of technology. And for us, the vertical integration of many of these pieces, I think, is going to lead towards greater independence and control and decision making. I think we also have to ask serious questions about what the actual impact versus the intent of this like large, just this large amount of funding that's going into genomic technology development. I mean, has it had the impact in health disparity and reduce that gap in health disparity that we'd like to see? And I would say on the whole, no, it hasn't. If you really spend time in a lot of indigenous communities or communities that don't have access to healthcare, there are different priorities, if you will, fresh water, et cetera. So I think right now we're building towards vertical integration of many of these pieces, and we're trying to empower our communities in that way. And we have some very exciting news that I don't think I'm allowed to talk about yet, but I mean, it's looking bright. So get your sunshades, you know.
All right, brilliant. Well, I'll stay posted. That's fantastic to hear. So maybe I'll now ask some broader questions for the panel as a whole. But maybe this question might be a bit more relevant to you, Professor Kenny. So there's a lot of, I would say, debate now in terms of the appropriate usage of self-identified race and ethnicity. Which some people, rightly recognize that, these are largely socially constructed measures. They don't perfectly capture kind of the experiences of people in different communities. You can think of Hispanics, which often have quite complex genetic ancestries admixed within their genomes. And that one label, Hispanic, doesn't necessarily do a good job of capturing it. And I've read your cell paper, which is brilliant, and you show that using the scale-free, network-based IBD approach for constructing communities results in these designations that are discordant from self-identified race and ethnicity. However, there are others that point out that as flawed as race and ethnicity categories are, they still capture some useful information. And there was a commentary in New England Journal of Medicine with Esteban Burchard as a senior author who said that it's still kind of a master variable that captures let's say discrimination and racist experiences that people might feel in other socioeconomic factors. So I'm just thinking going forward, what role should self-identified race and ethnicity play when we think about personalized medicine research?
Yeah, so it's a great question, and Esteban is a friend and colleague. And to echo him, what he would say, and I agree with this, you don't want to throw the baby out with the bathwater. In other words, decades of epidemiological research has demonstrated that there is valuable information in the variable of race and ethnicity, as imperfect as it is, because really what it's capturing is a lot of what we tend to, in epidemiological research and even in genetic research, just shove into one variable called the environment. And we know the environment isn't the same for everybody. And I don't think that race ethnicity is the best solution for that. But in some cases, it's the only solution to capture information that we know is impacting people's health. Now, having said that, I think we need to move away from it. I certainly think we cannot solely use a variable that we know is so imperfect. And I think we need to get a lot more nuanced and complex. Even the idea of thinking about race and ethnicity, it's a very US-centric, it's very, flawed in any aspect of its ascertainment. And a lot of how people truly self-identify is really depending on who's asking them and where they're being asked and for what reason. So I think that we do have to think about how we can better capture what we're really trying to study, which is what are the social, structural, environmental determinants that are impacting this person's health. And I think that we can use genetic ancestry to some extent. I also don't think it's perfect, but it's a lot more agile and flexible, and we can toggle it up and down in different temporal scales, and we can start to get much more sophisticated about our modeling. And I think we need to do a much better job of capturing those structural and environmental factors that are impacting health, and bring them into our biological modeling.
Yeah, no, brilliant. That's good point. And you know, there is quite a frustrating level of variability in how these ethnic categories are designated across countries. Asian in the United States means something different than Asian in the United Kingdom, and it's But
even one thing in the United States, as it doesn't also in the UK, right?
Keolu, Amy, is there anything else you'd like to say to that?
No, I think that's a really accurate pulse on the issue and the complexity that's going on. And I think it's going to be a very exciting next decade and future and so on. And we'll see what we can actually do.
Yeah, and to add to what Jason has just said at the end there, African in Africa, it's different than African in overseas. And we are all one community when we come out of Africa, but we are not a same community when we go back to the continent. What part of the things that we are trying to do with those facial studies, we are starting from the premise that identical twins, they have very similar faces because they share lots more of the genetic profile. So the more we tend to differentiate, the more we are also divergent on the genomic side, and the more we are different in terms of response to the environment and other factors that may affect our health. So even within the African continents, there are much more to study with regards to that in terms of the response to the environments. We are not responding to the same, we are not exposed to the same factors which might create some type of selections, and we are not responding in the same way. So very interesting topic to continue studying.
Absolutely. Amy, I noticed that you flagged two questions in the Q&A that you said you'd like to respond to, and I'd certainly like to give you that opportunity. Are these questions referring to the role that whole genome sequencing plays in Africa, or?
Yeah, I would like to start with the second one related to the phenotypic study. We did this study comparing Belgian Down syndrome patient to Congolese Down syndrome patient. This is only part of the results because we also compared Down syndrome from other African countries to show the differences that might exist between African patients and the distance that might exist with other patients from African ancestry that are living in the island of Guadeloupe. They have been originally from Africa with slavery trade and all this. We actually were able to prove that those populations are sitting in the middle between the African and the Belgian. So it shows similarities, and it's really support the fact that there is a baseline genotype that is influencing the baseline phenotype, even though this is not part of this genomic syndrome, but we need to take this into account when we are doing the description of the disease. If a disease is described for the first time in Congo, we would say that this syndrome is characterized by thick lips, by broad nose, which is not part of the syndrome itself. It's just part of the normal population where the phenotype or the disease has been identified for the first time. So we need to look at all those elements that are distorting the description of phenotype to be able to identify the core element of the phenotype. And doing these type of studies in diverse population, it's really helping. And we hope that much more people will engage and associate with us so that we can expand these experiences in other communities. And with regard to the genomic study, yes, there are lots of studies that are going on. The H3 Africa, with the H3 Africa chips that have been designed with Illumina, we have been able to produce lots of data that are being, for the moment, already interpreted. We are not waiting, they are already being interpreted. And from the first results, the idea is now to go further. We know we have a sense of the variations that have been observed, but we would like to get a sense on the impact on the health status. That's where the whole genome sequencing is really coming, because it helps us to have a clear idea on the structure of the human gene, the African genome, which are the gaps that we are not capturing, and how the structure of this part that we are not capturing may influence our health. So this is the way we are going with much more sequencing, even using long read sequencing to try to redraft the African genome. So it includes multiple countries where data are already collected. But as I said, two things need to be considered: the amount of data we produce and the diversity of data we produce. We not only need to sample more, but to sample much more broadly. In multiple countries, we are missing information. This is the effort now we are doing to involve more countries. As we are speaking now, there is a meeting of the H3 Africa where this particular topic is being discussed as we are speaking.
Brilliant. No, thank you. That's it's very interesting. And I guess just a related thought there, just maybe zooming out a bit, you touched on the cost of or whole genome sequencing and its role for providing more insight. I guess historically or to this point, to what extent has the actual cost of genotyping and whole genome sequencing technologies contributed to the health disparities that we're dealing with right now? And I guess this is maybe a broader question for the panelists.
Maybe I will just provide a short answer. The yearly average salary for someone that is working full-time employee in my country is a total about $800 a year. So this is less than the cost of sequencing. That's a yearly salary you need to survive. It means that it's really out of reach. It's impossible. What we have been able to obtain has been through projects, research projects, and for the moment, we are having much more collaboration with philanthropy. As Keolu mentioned, the Regeneron, Illumina, and other players are coming on the field really to try to help. But when this will be finished, we don't have a clear path, as our countries are not really involving themselves into this area yet.
Kalu, Emir?
Oh no, I could just add. I think we're there's that famous NHGRI slide, right, with the just drastic decline in pricing compared to Moore's law, right? Like that. I mean, so I think it's becoming quite ubiquitous, and there's all kinds of new technologies and perspectives, even long read sequencing. I understand Dr. Kenny is working on some really cool kind of pan assembly stuff. So I think we're totally moving in that direction. I just hope that our communities are in control of the data. They're the gatekeepers of who has access to this, right? So that's the kicker for me.
Well, exactly to both of your points, I think the consideration is not just the unit cost per sequence. Actually, it's all the infrastructure that goes around it. And there's a reason why most of the sequencing is done in these very centralized places that have, the infrastructure and the funding to actually support that kind of centralized endeavor. I do think it's true that the price of sequencing precipitously dropped in that famous plot, but then it flatlined for a couple of years. It didn't go down much past that flat line. I think that's going to change. I think there's much more competition on the market. There's new technologies, technologies built in not for the large machine centralized model technologies that you can wear on your back and go out in the field. Mm-hmm. So I think that cost will always be an issue outside of resource-rich environments, but the infrastructure around it, I used to be very, very compute-heavy it wasn't, I mean, it wasn't just the sequencing. You needed a whole team of bioinformaticians with the expertise in place to analyze data. And only, you know, but now I think that's really starting to become much more accessible and available and much more pipelined, and you know, tools are easier to use. So I'm hoping, and you know, we can do a lot actually, we know, with even very super low coverage sequence data. And also to Dr. Lumaka's point, chips that are actually tailored for the population that you're trying to study, where they have really good dual scaffold and reference panels. That model of chip and imputation, it's not going anywhere. It's still a very, very viable model. Why? Because you don't need big sequencing centers to do array data. Anybody can do array data. And so that's why that model didn't go away. Everybody thought it would, but it didn't go away. Chips, I think, are very powerful tools for research and even medicine, and you can do a lot with them.
Yeah, very, very good points.
Could I add, Dr. Torres? I totally agree with Dr. Kenny. I think that's the most thought-provoking aspect of it. It's building the infrastructure so that we don't force our communities to outsource biological information. So if you can, you know, create just a really high-res panel of variation that represents, it's harder than it sounds, right? We're trying to do this in Polynesia, and then you impute and model, and you can impute you can impute some awesome stuff, structural variation, a number of things. But yeah, I think it's very exciting.
Yes, these are all really great points. I'm conflicted because there's more questions I want to ask you, and it's such an expert panel, and it's so rare to have you know, people of this caliber to talk about, you know, these issues together. But we have we should take another break now. There's our final session with our keynote speaker, Dorothy Roberts, starts in 14 minutes. So I feel like we should let people get a little bit of a break. But you know, it's been an absolute pleasure to have you all join us today and hear your fantastic work. So Professor Kenny, Professor Foxx, Professor Lumaka, thank you so much for your time.
Thank you.
Thank you, everybody.
Cheers.